In this video, I will discuss two binary expression systems used in Drosophila, the GAL4-UAS system and the LexA-LexAop system. A binary expression system allows scientists to investigate gene function in an organism by providing the opportunity to knock out or increase expression of a gene in a tissue-specific manner. By preventing cells that normally produce a protein from making that protein, you can assess phenotypic effects associated with the loss of that protein. From here, scientists can draw conclusions about the protein's role in the type of cell under investigation. This system can also give scientists information about the spatiotemporal expression patterns of genes that they are investigating. The GAL4-UAS system is made up of two parts. The first piece of the puzzle is GAL4, a gene normally found in yeast. It encodes the transcriptional activator protein GAL4. This gene will be inserted into a fly genome, placed under the control of a native enhancer. So when this enhancer is active, GAL4 is expressed, and when the enhancer is inactive, GAL4 is not expressed. You should note that different enhancers are only active in specific tissues at specific times. The other part of this system is the upstream activation sequence, UAS. This segment of DNA is an enhancer that, when bound to by GAL4, promotes the transcription of a gene of choice downstream. This gene is called a reporter. So we start with these two elements, GAL4 and UAS, in separate fly lines. The driver line contains the GAL4 gene inserted into the DNA of a fly under the control of a native enhancer. This enhancer is tissue-specific. That means that it is controlling the GAL4 gene expression and is only going to activate transcription of GAL4 in specific types of cells of the fly, perhaps only in nervous tissue, or more specifically, a single type of neuron. The other line you will use is the reporter line. This line contains in its genome the upstream activation sequence and a reporter gene. When you cross these two lines, you get a generation of flies that contains both the GAL4 gene and the reporter gene. In these flies, the GAL4 will only be expressed in a tissue of choice where the native enhancer is active, not anywhere else in the organism. In the cells in which GAL4 is expressed, the GAL4 protein will bind to the UAS, triggering the expression of the reporter gene. This gene can be a variety of different things, depending on the goal of the experiment. So what can this reporter gene do for you? A common reporter is the gene GFP, which encodes green fluorescent protein, a protein that glows. Using a reporter line that produces a glowing protein allows a scientist to know exactly what cells are expressing GAL4. In the cells in which GAL4 is expressed, it will bind to the UAS, which will then promote the expression of GFP, producing a glowing protein, which is easily visualized in the organism. Conversely, in the cells in which the enhancer of GAL4 is not active, GAL4 is not produced, meaning the GFP will not be expressed. These cells will not glow. You can then directly isolate those cells in a machine called a flow cytometer that selects the glowing cells and purifies them from the non-glowing cells. In this way, you can use the GAL4-UAS system to find out when and where the enhancer that controls GAL4 is active. This gives you insight into when the genes that are regularly controlled by this enhancer are active in the fly. So why did we start with two separate lines of flies, the driver and the reporter? Once you know when and where the enhancer that's controlling GAL4 is active, you can cross your driver line with a different reporter that will allow you to investigate cell and gene function in those specific cells. You can do this by crossing this driver line with a new reporter line. If your driver line is under the control of an enhancer specific to a subset of neurons, you could cross it with a reporter line containing channel rhodopsin. This would allow you to control the firing of neurons with light. You could also use a reporter line that encodes shRNA, which would allow you to knock down gene expression of specific genes in the cells expressing GAL4. This would give you insight into that gene's function in that specific type of cell. There are many other reporter possibilities you can also use with this driver line. As you can see, mobilizing GAL4 in the genome under a tissue-specific promoter or enhancer allows you to then manipulate gene expression in that specific tissue to better understand the roles of the cells and the genes expressed in those cells. The LexA-LexAop system is quite similar to the GAL4-UAS system. In this system, LexA is a transactivator natively found in bacteria. This transactivator interacts with the LexA-op promoter just as GAL4 interacts with the UAS. That said, the LexA DNA binding domain is not able to interact with the LexA-op promoter in flies, so instead we use a LexA fusion protein called LGH as the transactivator to allow this system to work. So you place a LexA fusion driver line under the influence of a native enhancer. You then cross in a reporter line with the LexA-op promoter linked to a reporter gene. You now have another binary expression system. The reporter genes can be any of the reporters I mentioned earlier, GFP for identifying where the enhancer in the driver line is active, channel rhodopsin or shRNA for later manipulation. All of these reporters can be used with the LexA-LexAop system to investigate gene function in cells. So why is it necessary to have two binary expression systems available for use in the lab? Well, these two systems will not influence each other, meaning that the GAL4 transactivator will not interact with the LexA-op target, while LGH will not interact with the UAS of the GAL4 system. Using both of these systems at once allows you to study the interaction between two sets of tissues, one being manipulated by the GAL4-UAS system and one being manipulated by the LexA-LexAop system. In this way, you can manipulate the expression of one protein in one type of tissue while simultaneously manipulating the expression of another protein in a different type of tissue. These two systems open up a whole new realm of opportunity in the study of expression patterns and protein function in Drosophila. So those are the basics of the GAL4-UAS system and the LexA-LexAop system in Drosophila. See my video on enhancer trapping to learn how a P element can trap a native enhancer.